Historically, transcription factor activation studies have used gel shifts, immunoblots, and reporter assays, which are all time-consuming, low throughput, and only semi-quantitative. Now there's a better way. TransAM assays from active motif uniquely measure levels of activated transcription factors and are quantitative, up to 100-fold more sensitive than gel shifts, and can be completed in fewer than 5 hours. Here's how it works. First, add your cell extract to the TransAM assay wells, which are coated with DNA containing binding sites for your activated transcription factor. Next, add the primary antibody targeting the transcription factor, followed by a secondary antibody and developing end-stop solutions, resulting in a sensitive, quantitative readout. And TransAM assays enable researchers to screen up to 96 samples simultaneously, saving time and money. But don't just take our word for it. Our TransAM kits have been cited in over 1,000 publications. For fast, reliable results, upgrade to TransAM now.